Thank you guys for coming. I actually was really nervous that nobody would actually come. So this is about the CURE program that I did with the Dana-Farber Cancer Institute with Harvard. And it was a 3-week program, and I was fortunate enough to be working with a professor who had 3 experiments that she was working on at the same time, so I got to work on a little bit on each project for each of the 3 weeks. So, like I said before, this was done with the CURE program, and it's a program where it helps the minorities get into the science field and research. And I actually found this last year because this is my 2nd year doing this program. And how I came across it was I just did a simple Google search, and this was one of the first results that showed up, the CURE program, so I clicked on it. Unfortunately, it was only available for Massachusetts residents, and I came from Massachusetts, so I sent the people who were in charge of the program a little email saying I was really interested, and they deferred me to a professor, the professor that I worked with, Dr. Wang Xu, and she allowed me to, last year, she let me be a summer intern, and she invited me back this year for 3 weeks. And that was how I got in, and I settled the date topics through an interview. And before we get into the topics, I'm going to tell you about the terminology that's required to understand it, because I'm not sure if you guys would know what these words mean. So we'll start with the Western blot, which is a procedure that's used to detect and identify specific proteins from a cell culture. And that's one of the most important procedures that I had to do during my experience at doing research at the Cancer Institute. The second thing is the knockdown gene, which is a gene whose expression is reduced or suppressed by a protein or a reagent. A rescue experiment is an experiment that's not done to not an emergency experiment done to rescue the results, but it's actually an experiment to confirm the results of a previous experiment to show that the knockdown, the gene's expression, the reduced gene expression is due to the knockdown and not some other factors. And an exon is any part of a gene that holds information for RNA after RNA splicing. So after the introns are taken out, all there's left is the exons, and those hold the key information to gene expression. So like I said before, the Western blot procedure is the most important procedure that I had to do. And it's for seeing and identify proteins, and it's done with 7 big major steps. So the first step is to take your cell cultures and to isolate the protein, so you lyse the cells and break down the membrane so you can get to the protein inside it, and you suspend the proteins in chemicals and in a solution that contains chloroform. And after that, you have to check the protein concentration to make sure there's not too little or too much protein, because if there's too little protein, you won't get any results after the Western blot, and if there's too much, you'll just see a big blob on the page. After you check the protein concentration and make sure that it was the appropriate amount, you make the SDS page protein gels, which is used for gel electrophoresis. And so the proteins can separate by weight through the gel. And gel electrophoresis is when you run a current through the gel so that the proteins can separate. After you've done the gel electrophoresis, you transfer the gel results to a nitrocellulose paper, a special kind of paper that's used to, that allows proteins and antibodies to bind to it. So, and this is important because of the last step when you're trying to to visualize the proteins and to actually make it appear on paper. We can't use the gels to do that. So you transfer the gel results to nitrocellulose paper through another gel electrophoresis, where it electrically sends a current through the gel and the paper to make it bind to the paper. And then you have to soak the nitrocellulose paper with the proteins in dry milk and antibodies, so the antibodies can also bind to the proteins on the nitrocellulose paper, and that's actually how you can see the proteins afterwards. So after you put in the corresponding antibodies and the proteins together, there's a lot of different methods to being able to detect the proteins after on the nitrocellulose paper, because with the naked eye, you can't actually see or smell or feel any proteins. You have to use, what my lab used was, we used a radioactive detection method, which required X-rays, and you would perform an X-ray of the nitrocellulose paper, and then on the X-ray film, the bars and the bands would show up. And here's a picture of it. So on the left side, you can see that's the nitrocellulose paper when it just came out of the antibody and dry milk mixture. There's nothing on there except for the molecular marker, which is standard. And on the right side, you can see that the bands did show up, and the blue film is the X-ray paper. So this is what the Western blot actually looks like, the bands. So, like I said before, I did 3 research topics during my 3 weeks, each topic for 1 week. And the first one that I did was very, very basic, and it was to categorize the antibodies into a Western blot for the C2, C12, 4.1R proteins. And it was the rescue experiment for my professor. The second one is analyzing the analyzing the effect of caper A knockdown on the exon 16 splicing in the C2, C12 cells. And on the third week, I analyzed the effects of TIA1, PCBP1, and caper A proteins on the WTXM and UP mini genes. So the first experiment that I did was the rescue experiment, and the C2, C12 is a cell culture of muscle cells that was extracted from a few cancer patients that agreed to testing. So what we did with these cells was that we introduced the 4.1R protein into these cells through transfection, and we let it incubate for a couple of days and replicate itself in the cells, and then we soaked the cells in doxycycline reagent to induce a knockdown and to suppress the genes. And then what I did after that was I did the Western blot procedure to the cells to see how well or how badly the expression showed up in the genes. So this is an image of the different transcription factors, and this is the Western blot results that I got from the Western blot that I did, and the plus sign on top, you can see the pattern of the plus, minus, plus, minus. The minus sign represents that there is no doxycycline, the knockdown reagent, and the plus sign indicates that there is. So as you can see, that the experiment was a success, because when there was no doxycycline in the cell or in the proteins, there's a much clearer band that shows, and with the doxycycline there is not a very clear band that shows, which means that the reagent doxycycline did knock down the expression of the genes. The second experiment that I did was the effect of caper A, a protein, and how it knocked down the expression of exon 16. And like before, I transfected the cells with the caper alpha protein, and we had to do RNA isolation because the exon is expressed in RNA. So we isolated the RNA from the transfected cells and extracted protein for the Western blot procedure. And these are the results. The control means that there's no caper A, and the caper A means that the control shows that there is no caper A, and the caper A means that there is the protein in the cells. And caper A is supposed to, it was a knockdown, so that means that it suppressed the expression of the gene. And as you can see, the bands that are in the control with no caper A are a lot more darker and vibrant than the bands with caper A, meaning that caper A did its job and suppressed the genes and had a successful knockdown. The last experiment that I did was during my 3rd week, and it's still an ongoing experiment with the laboratory at the moment. So it's still like an ongoing, so I'm not sure what the results are. I haven't gotten like any word back from a professor about what they actually found. But this was, I just tested some of the hypotheses that I had at that point. And the 2 hypotheses were that the TIA1 protein, the PCBP1 protein, and the caper alpha proteins would enhance the caper 16 expression. And because even though, like previously, we saw that caper A actually knocked down and suppressed the exon 16's expression, with the help of TIA1 and PCBP, it could actually enhance the expression. So what I did with this experiment was that we used MEL cells, which is leukemia mice cells, and we introduced different combinations of the 3 proteins into the cells, and we isolated the RNA for each of the cells to perform PCR with the RNA. And this is the RNA results and the Western blot results. And as you can see, this is the most important part. And the top one on the black, the bottom band of the 4 black bands, are the most important because it shows that that there is a solid, there is a very strong expression of exon 16 even with the presence of caper alpha in the cells because of the 2 proteins, TIA1 and PCBP. And on the second side with XM and UP, those are mutations of the TIA1 and PCBP sites. So when we introduced these 2 mini genes into the MEL cells, it inhibited, it prevented the 2 proteins to successfully bind and do its job in the cells. So as you can see, the V represents that, shows that the XM and the UP mini genes were in the cell. And there's no, as you can see, there's no band shown here, which meant that there was no expression because those 2 proteins couldn't do its job, and the caper alpha suppressed the exon 16 expression completely. And so those were the 3 experiments that I did, and it was a really cool experience because I put my AP biology knowledge to the test when I was there. And I worked the schedule of a typical researcher, which isn't the normal 40 hours that you would think of when you work in a lab, when you would normally work. Because as a researcher, I've learned you have to be willing to get, like for me personally, I had to go in on Saturdays and Sundays, so it was a 7-day work week, not just a 5 days, because the experiments that I did required me to incubate cells overnight or for 12-hour periods. So I had to go in on maybe an early morning Sunday or really late at night on Saturday to check on the cells and to keep the procedure going so nothing would go bad. And working with my professor who was doing the 3 experiments at a time, I saw how to get grants and how to commence an experiment and to start writing a paper. And it's a really long process. I remember my professor was always really busy in her office, like trying to get all of these grant proposals done, because when you get a grant, it's very important that you start and you actually use the grant, and these grants can last up to 4 years. So you also have to be very dedicated to the topic that you're researching on, because these grants last a really long time and you really have to make good use of the money that you're given. And lastly, at the end of my 3 weeks, I was able to meet the president of the Institute, Dr. Edward J. Benz, and present my findings and my experiments to him. And it was just me, my professor, and him, and we got to sit down at a table, and he just looked over my findings and then approved them to be sent off for publication. And that was basically my experience at the Dana-Farber Cancer Institute, and thank you. Does anyone have any questions?
What was the most challenging part of this whole experience for you?
It was really about understanding what was going on, because as you could probably tell, it was really intense, and it was talking about some things that I've never actually heard of. So during my first week, my professor gave me about 3 really long scientific papers that she asked me to go through. And half of the papers I really couldn't understand, so I had to sit down with my professor one-on-one, after my hours, to actually talk to her and get to understand what all of it meant, so I could perform the experiments correctly.
So, are you considering a career in this kind of research?
Yeah, I am. It really opened my eyes to see that a career in biology doesn't always have to be a medical doctor or something to do with or just chemistry or engineering. It just because before this I never really had thought about being a researcher, but after my experience here I really enjoyed it, so it's definitely a possibility.
Did they incorporate you as part of their team on an equal basis?
For the most part, yes, but there were some times, for the last experiment, I was really confused, so my professor really had to take me step by step for each step to really explain to me what everything was and how everything worked. Anyone else?
Thank you so much.